everybody. In this video, I'm going to talk about acellular pathogens, particularly the viruses and the prions. When I say acellular pathogens, we're talking about disease-causing microorganisms that aren't made of cells. And I'm going to talk about section 5.1. So in 5.1, the first objective on your lecture study guide says explain what it means when viruses are described as filterable. And this actually is leads us back to how viruses were discovered. And here we have 1892. Oh, this is during the golden age of microbiology, when when a lot of scientists were trying to figure out what microorganisms were causing what disease. And one of the common things that people did is they took tissue. In this case, there was this disease in tobacco plants called tobacco mosaic disease. Now that it's still around today, and the scientists of the day wanted to know what causes this disease. Is it caused by microorganisms? And at that point, they knew a lot about, or they were starting to learn a lot about bacteria. And they had techniques for isolating bacteria. So one of the things what they would do is they would take tissues like this, infected tissue, and grind it up and squeeze out the liquid, and that would release the bacteria in there. And so they'd have a solution that had the pathogens in there. And then to isolate the bacteria, they would pour it through a filter. So here's this is a little funnel, and it's got a filter right here, and the filter holes are small enough to trap bacteria. And then what scientists were doing in those days is they would take the bacteria from that filter and they would grow it up, and they'd try to link it to the disease that it was causing. Well, what happened when they did this with tobacco mosaic disease, they ground up the tobacco leaves and they filtered the liquid, and they didn't find any bacteria that caused tobacco mosaic disease. But what they ended up doing is checking the liquid that went through, and in that, they actually found something that caused this disease. So you could take the liquid, expose healthy plants to it, and they would become diseased. And so the very first thing that, it turned out this was caused by tobacco mosaic virus. And the very first thing scientists learned about viruses is they caused infectious disease, but they were smaller than bacteria. They could go through the holes in those filters. So very first things we knew about viruses as pathogens is that they were filterable. They were small. But there's all sorts of other things we've learned since then. And here's a table from your book, and I want you to list the properties of viruses that distinguish them from other microorganisms. And there's a lot of things listed here that are all really cool, and I'm just going to point out a few that really define viruses. And the first bullet here says they are obligate intracellular parasites of bacteria, protozoa, fungi, algae, plants, and animals. All living things are able to be infected with viruses, even bacteria. So, and what we mean by obligate intracellular parasite means that the virus can't survive outside of, it can't reproduce, it can't function normally outside of a host cell. So they have to live inside of those host cells. I kind of think of viruses like having teenagers or having kids. The kids, they don't own a couch. They don't have their own TV. They don't buy their own food. Their parents do, or their family does that for them. And so I think of kind of kids as a model, the kids in the family as a model for viruses living in the host cell. The host, the family, the parents, provide the kids with everything they need. So the kid doesn't need anything on its own. The kid has to live among the family. They're an obligate intracellular parasite. Couple other really defining characteristics. One I already said, they're ultramicroscopic. They're much smaller than bacteria, and I'll show you a picture related to that. A big one is they're not cells. Their structure is very compact and economical, but they're not made of cells. here it says they do not fulfill the characteristics of life. And I'm going to show you something on the next slide related to that. We're going to talk about structure, but one of the interesting things about structure is that they have to have nucleic acid in order to have the instructions on how to make the virus. But they have DNA or RNA, but not both. So you might remember from other organisms, we have DNA, we make RNA, and that's used to make proteins. But viruses don't have DNA and RNA. They have DNA or RNA and not both. And that DNA or RNA can be double-stranded DNA, which is typical, single-stranded DNA, single-stranded RNA, or double-stranded RNA. So viruses have really used the variety in nucleic acids to an extreme. Here it's telling you that they lack enzymes, they lack machinery for synthesizing proteins, and these are really falling within the characteristics of life. But if I had to, you had to really just say, how are viruses different than other pathogens? I would definitely say they're obligate intracellular parasites, they're very small, they're not cellular, and their nucleic acid is DNA or RNA, but not both. I put this here, I'm using this to get to our next objective. And it says, explain why it's more accurate to describe a virus as active versus inactive rather than living or dead. And so we're saying, is that virus able to infect? Is it an active virus? But some people might say, is it a live virus or is it a dead virus? And so the objective here is saying, why is active versus inactive better terminology than living or dead for a virus? And it really comes down to this diagram here. This is a diagram of what we would call the characteristics of life. Humans made this definition. What does it mean to be alive? And we said an organism has to grow and develop, it has to reproduce, it has to have genetic material that it passes on and evolves. It has to be able to maintain a metabolism, a constant environment inside of its body, inside of its cells, in the form of homeostasis. It has a metabolism for to get energy and to use and produce energy or extract energy. It's made of cells. It responds to the environment. When an organism has all of these things, we say it's live. In fact, we would say it's an organism. It's alive. But when we look at viruses, viruses really only do a couple of these. They do have genes and heredity that they pass from one generation to the next, and they can evolve, and they do reproduce. But other than that, all the rest of the characteristics of life are not consistent with viruses. Again, if I go back to my analogy of kids living with their families, the virus doesn't need a metabolism. It doesn't need to maintain homeostasis because its host cells are doing that for them. So they rely on their host for making their proteins, for making their any kind of energy, forms of energy that they use. And really viruses only occupy a couple of these characteristics of life. And so rather than saying they're living or dead, you're really better off saying it's an active virus, meaning it's able to infect a host, or it's inactive. And we might use that terminology to say, how long is a virus active on a surface? When we were in the COVID-19 epidemic, a lot of what we were worried about is how long can virus remain active on a surface so someone else can come by and become infected? And at what point does it become inactive? So it's important to know that about viruses. I want to talk just a little bit about how viruses are named. And the viruses used to be named by the diseases they caused. We'd say like the polio virus or the scarlet fever virus or the measles virus. And the difficulty with that is people might call diseases different in different parts of the world, or and so a lot of times people were talking about the same viruses and they were calling them different things. And so we realized that we needed some organization to keep the names of viruses consistent. And so there was developed the International Committee on the Taxonomy of Viruses. I think it was in like in the 1970s, I'm pretty sure, somewhere around there, where we just said, let's get all this straightened out and make sure everybody, when we're talking about a virus, we're all keeping the same names. And that what they ended up doing is they grouped viruses by their structure, by their chemical composition, and their genetic makeup. And so there's a lot of details that go into how viruses are grouped together. But at the very least, I want you to understand that there are ways that we can put viruses, similar viruses together when we're talking about them. And when we're talking about what virus causes polio, the polio virus, we know everybody's talking about the same one. The you, Again, during the COVID pandemic, it you there was a lot of discussion about naming the virus and naming the variants. And so it was happening so quickly. I thought it was really interesting to see a variant name. Somebody would come up with the name of a variant, and then quickly, or maybe not so quickly, there would be kind of a decision on how do we name these variants, or there would be the names might actually change because the ICTV came up with a naming system that worked a little bit better, or that was a little bit more consistent. So it is something that is important for us understanding about how viruses are named. Here's a diagram from your book, and you can look at it. It's table 5.7. But what it is, it's just showing you that, they have it divided into two groups here. Here's DNA viruses and RNA viruses. And then they grouped, they have a genus name they have here we go. They have a family name and a genus name. They don't really have species names like we do for bacteria and all other organisms, but a lot of viruses, or most viruses, will also have a common name. And so here we go. The coronavirus is the genus name, and the common name of that virus was SARS-CoV-2, and that name was developed by the International Committee on the Taxonomy of Viruses. The disease it causes is COVID-19. So you can see a lot of viruses that you might have heard of before here. That's all I have for section 5.2. You can go to the next video to see the video on section 5.3.